Hey students, it's Professor Fernetovich here. This is part 2 of our Cannabis and Genetics lesson. We have been talking about Mendelian genetics and crossing. We've used some terms like alleles and gametes, P, F1, F2, to talk about genetics. And so I left off giving you this slide and having you look at it, giving you some time to just glance at this. You can even pause now if you didn't get a chance to before, because I want to walk you through this example of a monohybrid cross. And so if you remember back to our last lecture, a monohybrid cross is where we're looking at one trait. Okay, and we have a P generation here, and these are true breeding plants that have always shown these specific variations of the trait. We're looking at height in this example. And this is an example from a bio textbook. We've got tall plants on the left and short plants on the right. So this has letters down here. These are the genotypes or the alleles. Capital, so dominant. So tall is dominant. And this little short plant here has two lowercase letters, two recessive alleles. So when we breed these, the tall plant can only donate tall alleles, those capital T's. It doesn't have any lowercase t's to donate. And the short plant can only donate the lowercase t's, the recessive alleles. And so this line is showing what the gametes are. So a sperm or an egg cell would just have tall genes, and a sperm or an egg cell over here would just have short genes. So when they combine, in the process of fertilization, when that pollen meets the ovum, we end up with a diploid zygote. That means it's got two alleles again. Remember, these are haploid gametes. They combine to make a diploid zygote, and it's got one of each, a tall gene from one parent and a short gene from the other. And because it shows that, because it has that big T, that dominant allele, that capital letter in its genotype, it's going to show the dominant trait. So that's what we have here, a dominant allele, a dominant trait for our plant. Pretty cool. But it's still carrying that recessive allele. So what will that do? Well, in that plant, that F1 generation plant, it's not going to do anything. But if you breed F1 generation plants together that are what we call heterozygous, have one allele of each, you get something different. And that's shown here. This is a big Punnett Square. Well, it's not that big. But this is what a Punnett Square looks like. And this one's even a little bit complicated. I usually just draw this box here and partition it into fours. But we've got these columns up top and on the side. Well, what this is doing is taking the F1 generation and imagining breeding another one to it. So a big T little t bred together with another big T little t. So you've got a mom plant, a mother plant, shown here with the little female sex sign, and that can donate a big T from right here and a little t. So one each of those different alleles. They're different. And the father plant, with the male symbol right there, would be the same, and it would donate a big T and a little t as well. So what happens when we breed these two? We're actually breeding two heterozygotes together, and we have differences in what the genotypes are of the offspring and what they will actually look like. So when these two alleles would combine, we would get two big T's, if that was the case. Now this is all going to be random in what actually happens in the plant, but what we're doing here is looking at probability in our offspring. What is likely to happen? What's the most predictable thing that will happen when we cross two heterozygotes? Well, big T is one possibility. We can get a lowercase t from one parent here and an uppercase T from the other parent there. We get one of each, so we get another heterozygote, same genotype as the parents. Down here in this square, we take the big T, drop it down, the little t, bring it over, they combine. In that situation, another heterozygote. Now these two are genetically similar, even though they're getting the lowercase t and the uppercase T from different places. They still end up together, and it's standard practice to put the capital letter before the lowercase letter. It doesn't really matter. It shows that we have two different alleles. We've got heterozygotes. Well, down here in this corner of the Punnett square, we have two lowercase t's, one from this parent and one from this parent. So there's a chance of getting a short plant. That would show the recessive trait. So we have three different types of genes. We've got homozygous dominant right here, two of the same, and they're both dominant. We've got two heterozygotes, and then we have one homozygous recessive. So three different possibilities for our genotypes and two possibilities for the phenotypes, or what the plant looks like. So these heterozygotes show the tall trait, okay, because they have a dominant allele, and then this one has the recessive trait because it has two recessive alleles. So we have a phenotypic ratio that's shown here, 3 tall to 1 short possibility. So if you're doing a breeding experiment with heterozygotes, you can assume that this is going to be your offspring, what you will see in the next generation. Doesn't always work out that way. It's like rolling a dice, but this is the likely outcome. Pretty cool. So I used the term genotype and phenotype in that last slide. The genotype are the letters representing the alleles for a specific trait. And the phenotype is the physical appearance of the individual, what the organism looks like. Phenotype can be broader than that, though. It's not just what you look like, it's what your genes are expressing. So in terms of our cannabis plants, we could be talking about tallness versus shortness, or the color of the leaves, whether we have purple leaves or greener leaves. But we could also be talking about genetics involved in cannabinoid content. So percentage of THC or CBD, or what types of terpenes our plants are producing. So that term phenotype can be more than just looks. It can also be metabolism and chemical composition of our organism. Here's a Punnett square that I like to show in my general bio class of parents and ears. So this isn't cannabis related, but it gives us another practice. Here, hanging down earlobes are dominant to earlobes that are attached. And you might have noticed those different earlobe types in humans. It's not to say that hanging down earlobes are more common, even though in my experience I've seen that they are. It is that if you have hanging down earlobes, you have a dominant allele in your genotype. And you might have two dominant alleles, being homozygous dominant, or you can be heterozygous, like what's shown here, with one each. But that dominant allele shows. So this is looking at something just like the F1 to F2 generation cross in that previous slide. We're breeding two heterozygotes together. And so from this parent, let's say this is the female, she can donate the capital E, the dominant allele, or the recessive allele. Okay, they split apart when the gametes are formed. So half of her eggs have the dominant allele and half of her eggs have the recessive allele. Same with the male parent over here. He's got capital E, a lowercase e. Half of his sperm have the dominant allele, half of his sperm have the recessive allele. Imagine like a million sperm and a few eggs. Half of them have dominants, half of them have recessives. What's the probability and how are they going to come together? Right? So there's the possibility that two dominant gametes, the two dominant alleles will come together. We get a homozygous dominant individual, which is different than the parents, right? That's pretty neat. Or you can get a recessive allele from one parent and the dominant allele from the other. Okay, pull that over, bring that down. Or you can get dominant allele from mom, recessive allele from dad. Got a heterozygote here. Or you can end up with the little e's from each of them, with two recessive alleles there. So that's homozygous recessive. So just like before, we have dominant, dominant, dominant in terms of phenotype, what they look like, and a recessive. So a 3-to-1 phenotypic ratio is shown here. Even though we have different genotypes in our dominant phenotypes, they're still going to show the dominant trait because that dominant allele is present. So let's look at a cannabis example. Oops, I see a typo. Sorry. Say we have purple leaf color, like in grape soda strain. It's dominant trait over green leaf color. And we're just using this as an example. Purple, therefore, would be labeled a capital A, and green would be a lowercase a for our alleles. So a purple plant would look purple with one purple allele, since it's dominant, but it could have two purple alleles. So we could have big A, or big A, little a here. Homozygous dominant, heterozygous for a purple plant. We might not know what it is. We could do some back crossing experiments for that. You'll see that in the Cannabis Grow Bible. Or you might be coming from a true breeding long line of purple plants. So that would be likely homozygous dominant. The green plant, showing only the recessive trait, doesn't have any dominant alleles, so it must have the two recessive alleles and be homozygous recessive. What would the offspring look like if we bred a purple heterozygous plant, two different alleles, with a green homozygous recessive plant? What would the possibilities be for our offspring? So we're going to bring in a Punnett Square like this. The first plant is heterozygous, so we put a big A and a little a on one side of the Punnett Square. The other one's homozygous recessive, so we have little a here. And can you fill that in? Pause this video and do your Punnett Square. Try it out. Okay, I hope you tried that out. And I hope your Punnett Square looks like this. We would have two of the offspring being heterozygous and two of the offspring homozygous recessive. So this is different than breeding two heterozygotes together. We have half and half. Half would show the dominant purple trait and half would show the recessive green trait. So half purple, half green in that example. So we're just about out of time on this video, but you can see some more examples of this on pages 565 to 569 in the Cannabis Grow Bible. That's in chapter 16. And like I said at the beginning of the presentation, the Hardy-Weinberg part of the chapter is not important for us right now, but the Mendelian crossing is what you will probably have experienced the most in your cannabis grow. So pay attention to that. And as always, send me an email if you have any questions, and check out the Moodle site for more information on this topic.